Well, hello everyone, and welcome back to Science in the Age of COVID-19. Today we are super happy to be joined by Professor Lachlan Coyne of the Doherty Institute and the University of Melbourne. As you can imagine, quite a time difference between us and Lachlan, so he's being a real sport and joining us at his 6 a.m. So yeah, hopefully you've had enough coffee. Lachlan's lab has done a lot of great work on a lot of core bioinformatic tools, the 1,000 Genomes Project, the PFAM, Protein Structure Family Database, tools for GWAS analysis and digging into specific use cases of that, and more recently establishing a set of tools to use for long-read sequencing analysis. Lachlan's lab turned their attention to SARS-CoV-2 and the transcriptional and epitranscriptional analysis of cellular infection. So Lachlan, take it away.
Okay, well, thank you very much for the invitation to speak in this seminar series. It's a real privilege. Yeah, I'm really looking forward to sharing with you all the work we've been doing to try to understand transcriptional changes in SARS-CoV-2 using direct RNA sequencing. So I'm sure everyone is extremely familiar with the epidemiology of SARS-CoV-2. But just to go through it a little bit, it was first discovered in China in 2019 and obviously spread all across the world and has generated enormous social and economic burden. So that's on the negative side. There has been some positives though. And one of those, particularly from a research point of view, is that there's an increased focus on the value of biomedical research. And particularly recently with the development of the vaccines, there's an increased interest in RNA in the general public as a molecule and what that can do for medical purposes. Probably don't want to be reminded of the cumulative number of cases is enormous, obviously, across all parts of the world, and it has led to a substantial number of deaths. And this was as of 6th of June 2021, up to 3.7 million deaths globally. And of course, it's continuing to spread. And one of the reasons it's still spreading rapidly is the emergence of new variants. So WHO has classified several variants of concern, which I'll mention here because we look at these later in the talk. So there's obviously, they've renamed these, so alpha, beta, gamma, and delta are the main variants of concern, and they've arisen in different parts of the world. They're basically thought to have arisen probably in immune-compromised patients where they haven't been able to clear the infection. There's been a number of mutations that have been able to accumulate in those patients. And currently, delta seems to be the most transmissible variant. There's also a number of other variants of interest, which are also ones to keep an eye on. So as I'm sure most of you know, SARS-CoV-2 is it's an RNA, positive strand RNA virus from the Beta Coronaviriddae family, approximately 30 kilobases in length, and it uses the spike protein to bind the angiotensin converting enzyme 2 receptor, followed by cleavage from TMPRSS2 to get to infect cells, and this allows infection of hosts. So our interest in this, because we've obviously done a lot of long-read sequencing before, was applying long-read sequencing approaches to try and understand how the virus works. And so this is a schematic of the transcripts and genes present in the SARS-CoV-2 genome. And so at the 5' end of the genome, you have so-called leader sequence of approximately was the first 60, 65, 70 base pairs. And at the end of that is a TRS sequence, which facilitates the formation of subgenomic mRNA. Following that, we have on structural proteins, we've got ORF1A and ORF1B, which can be translated into protein directly from the molecule. But because it's such a long molecule, the translation machinery wouldn't get to the 3' end efficiently. And so SARS-CoV-2 employs a trick whereby it produces a series of nested subgenomic mRNA, which consists of the leader sequence from the 5' end joined together with RNA from the 3' end. And so you see that you get these series of nested products of structural proteins, so including the S380 and so on and so forth, being produced. And I should say this molecule, the overall molecule, is polyadenylated as well. And the fact that it's polyadenylated makes it ideally suited to use of direct RNA sequencing technologies from Oxford Nanopore Technologies. So the use of those technologies require that you have, on RNA, require that you have a polyadenylated molecule. So it's quite a good fit with studying this molecule. Of course, you can use other technologies as well. The benefit of long-read sequencing is you actually can better elucidate the content of the subgenomic mRNA. So how does SARS-CoV-2 produce this subgenomic mRNA? So it uses a process called discontinuous transcription, which occurs during the replication of the molecule. So as you recall, I mentioned it's a positive sense single-strand RNA molecule. So in order to replicate, it has to first produce the negative strand, and in the process of formation of that negative strand, it's thought that due to the presence of these transcript regulating sequences, which this yellow bars here, we've got the one on the left I mentioned, which occurs at the end of the 5' leader sequence. That shares microhomology with the other transcript regulating sequences, which occur immediately upstream of the start codon of the structural proteins I mentioned in the previous slide. And that microhomology, probably through the process of formation of secondary structure as well, means that as the molecule is producing the negative strand, it sometimes jumps from one of these TRS in the 3' end to the leader sequence at the start and produces a negative sense molecule, which has the middle bit missing, if you like. So it just has the leader sequence at its 3' end now, and the 5' end being one or more of these structural proteins. And then from these negative sense subgenomic templates, it then can produce copies of the mRNA for translation into protein. So in this talk, I'm going to go through mostly some of the results that we generated for this paper, which was led by very talented grad student in my lab, Jessie Chang. So as I said, long-read nanopore sequencing is very well suited to study of the transcriptional dynamics of SARS-CoV-2. And so we wanted to see how the transcriptome changed, particularly production of the subgenomic mRNA, throughout the course of a cellular infection model. So we used 4 different cell types. So we used the ones indicated here. The most commonly used one initially, at least, was these Vero African green monkey kidney cell line, and that's used commonly as a model for growing up viruses. It's kind of deficient in its interferon response, and as a result, you can grow them up to quite high levels in Vero cells. So we used that initially, and we also moved on to using some human cell lines as well. So the CALO3 cell line is a epithelial model, and the CACO2, it's human kidney cell model. And We also used A549 cells, which unfortunately we were not able to infect those effectively, so we didn't include those in the paper. And then applied long-read sequencing and at number of time points and looked at the differences and what changed during the infection process. So a bit more about the methods, and this was with huge amounts of collaboration from Cantor Subbarao's group, who infected the cells for us in PC3 facility. So Jesse, the grad student in my lab, grew up these cells, as I mentioned before, to 80% confluency, and which were then infected in the Superbug lab with SARS-CoV-2. And then over a course of infection, we took out RNA at different time points during the host pathogen during the incubation period. And then we applied a variety of sequencing approaches, all based on nanopore. So as I mentioned, direct RNA sequencing was our main interest because the really nice thing about direct RNA sequencing is you're sequencing the actual molecule, RNA molecule itself, without first converting to cDNA. And so you're avoiding, in principle, many of the biases which might occur during creation of cDNA. And there's also potential of measuring modifications to the RNA itself. So we did that. The downside with direct RNA is you get lower yield. The molecule translocates through the pore on the nanopore sequencing device about 7 times slower than cDNA would. And as a result, you get about 7 times less data, if not a bit more. And so it costs more to do. And also it's harder to multiplex replicates and multiple samples on the same flow cell, and you wouldn't want to because you don't have enough yield anyway. So it is a more expensive approach. So as well, in order to get replicates, in order to be able to do things like differential expression, we also did direct cDNA, and that's a process by which you can convert these long RNA molecules into cDNA and then sequence them, as well as PCR cDNA, and another approach which involves more PCR. So we applied all of those approaches to the multiple time points that we had collected and the different cell lines that have been infected. Another thing we wanted to do, so as I mentioned, one of our aims was to be able to run differential expression analyses. So in order to make sure that those were well calibrated and we had confidence that we were able to accurately estimate the abundance of the transcripts in the cells we were looking at, we spiked in some synthetic genes as internal RNA controls. So we used the sequins, which have been developed, which is essentially based on in vitro transcription of an in silico chromosome. So this has, I think it's about 8 different genes, and then each gene has a variety of spliced isoforms. And so they're an ideal spike in because we can estimate how well our methodology is estimating both the total gene abundance as well as the isoform specific abundance as well. And this in silico chromosome doesn't share well, it's very low homology with actual real life DNA and RNA. So it's actually quite easy to distinguish these, and we can then use them in parallel, so in the same sequencing run, to check that our methods are working well. After the sequencing, one of the first things we looked at was what proportion of reads were mapping to the viral genome as opposed to the host genome. And so that's indicated here with the lines. So these dashed lines. So we've got on the left-hand side the results from cDNA sequencing, which typically have higher yield and replicates. And the right-hand side, the results from the direct RNA sequencing. And then in the top row is the CACO2 cells, and then the CALO3 and the Vero cells down the bottom. And so the dashed line at the top of each of these indicates the proportion mapping to the host at 2 hours, 24 hours, and 48 hours post-infection. And so you can see, obviously, as you might expect, at 2 hours post-infection, it's almost exclusively host. And then at 24, we start to see this bottom line starts to increase, particularly in the Vero cells. So you can see in the Vero cells down the bottom, the infection kicks off very quickly. much quicker than in the human cell lines, and the percentage mapping to the virus increases at 24 hours to about 75%, and then decreases slightly at 48 hours. And so you can see the CACU2 cells infected pretty well at 48 hours, and the infection rate in CALU was more modest. Also on this slide, and it's probably been distracting, is the colors, which are the bars in the background. So as well as seeing what reads map to the viral versus to the host genome, we also want to see what proportion of reads were capturing the subgenomic mRNA. And so subgenomic mRNA reads should include the leader sequence at the 5' most end of the SARS-CoV-2 genome, as well as the 3' UTR and the poly A tail at the end, if they're formed by this process of negative strand disjunction, as I discussed earlier. So that's indicated here by the the purple bars, right? So the purple bars are the number of reads, the abundance of reads in transcript per million mapped viral reads, which include both the 5' and the 3' ends, which is subgenomic mRNA, or potentially full-length RNA genome as well. They're quite rare. And so we see that that also increases, as you might expect, during the course of infection. And we get about half of the reads mapping to the viral gene coming from the subgenomic mRNA. As well as the subgenomic mRNA, we have the things which include the 3' end, but not the 5' end. And so one of the potential problems with direct RNA nanopore sequencing, if you don't do any 5' capping, is that the molecule might just not sequence completely to the end. So some of these non-5'3" molecules might be things which started sequencing at the 3' end and then just didn't find their way all fully to the start of the molecule, or they could be degraded products. I'm not really sure. And then we've also got this other category, the non-5, non-3, which doesn't include the 3' end and also doesn't include the 5' end. So they're just from somewhere in the middle of the genome. And in principle, we shouldn't sequence those with this technique, because we're only sequencing molecules with a poly A tail, and I'll dig into those a little bit later. So once we've sequenced the reads and figured out which are viral mapping and which are host mapping, we then want to look at what are the subclasses of transcripts that we see in our mixture. And so we approach this as a transcript clustering problem. And so we generated, well, we plan to generate a tool which basically took the reads as they come out of base calling and could then cluster them into reads which came from the same transcript isoform, if you like. So reads which came from the same class of subgenomic mRNA, which would then go on to produce the same protein. And there's a couple of reasons why you might want to do this. One is simply to be able to count the number of reads from each class of subgenomic mRNA. Also, you might want to generate a high quality transcriptome for future work. And also for the RNA modification work that we had in mind, we wanted to be able to say what the RNA modifications were on specific different classes of subgenomic mRNA. So to do that, we'd need to be able to cluster the raw data from reads which came from the same transcript isoform in order to build up enough sequence to be able to carry out that RNA modification analyses. We also wanted to make this program, fast and not to just be able to work in a single pass of the FASTQ, ideally, and ideally as a streaming process, and something which ideally would not just be applicable to virus transcriptomics, but also be able to help us do similar sorts of things on the host side. So to fulfill that, we worked for a while on this program, which we've called MP Transcript, which essentially you should think of as a transcript clustering program. So its goal is essentially to take this input pool of nanopore reads coming off the machine, which consists of host and virus, and to first figure out which are host and which are virus, and then based on mapping to either the host or the virus sequence, identify basically just from the cigar string, which indicates the positions where it maps to on the reference genome. So it's like indicates where the exons are, if you like, and based on that, be able to assign each read on the fly. So without then going back and reclassifying. So we wanted to do something which could do it on the fly, assign to a transcript cluster, and then therefore enable these downstream analyses, including things like, so obviously we want to look at the breakpoints, particularly for the formation of subgenomic mRNA, to be able to look at the cluster depth for each of these categories of subgenomic mRNA, to be able to look at, to be able to get high quality consensus calls as well, in order to be able to look for things like deletions or insertions, which might, specific to these subclasses of subgenomic mRNA, and also then to be able to do methylation at the level of each subgenomic mRNA. And so the way we tackled this is kind of very simple in the end, which is just basically transform each cigar string into a numerical hash, which basically just records where the start and end of each of so where the end of the 5' mapping section is and where the start of the 3' mapping section is, if you like. So each of the breakpoints, such that reads which have the same numerical hash should come from the same isoform. And we also made that annotation aware. So going back to the sequin analysis that I mentioned earlier, so as I said, we spiked in sequins. So we could apply this tool to the sequin genes that we had spiked in. And the nice thing is we know what the specification of that sequin mixture is. So we know what the isoforms are and what the relative abundance of those isoforms are. So we can see whether our observed log-fold change, for instance, between, I should say we spiked in, there are two sequin mixes. There's mix A and mix B. We spiked in the mix A into the infected and the mix B into the control, so that there was and they have different levels of abundance of the isoforms. So we could see whether our observed log-fold change calculated from the bioinformatics tools we applied matched the known log-fold change. You could see that it matched very well. And we could see for each of those mixes whether the observed abundance matched the known abundance. And again, you can see that actually worked quite well. So the tool's doing a pretty good job of figuring out gene abundance here and also isoform abundance. So now that we had a tool which could cluster reads into isoform classes, we could apply that to our data and look at what was the most abundant isoform cluster we were seeing. So this is just looking at all those reads which we labeled 5'3", which maps to both the 5' end and the 3' end, so which are genuine subgenomic mRNA. And so this is an example of the labeling that we gave these transcript clusters down the bottom. So you see the first one says leader _ leader and then n _ end, which just means that it's the read maps at the start to the leader sequence and at the end of the first 5' junction to the leader sequence, and then has the comma indicates that there's then a discontinuous gap, and then it maps to the just upstream of the N off, and then continues on to the end of the SARS-CoV-2 genome. So this transcript would then go on to produce the N protein, and we can see that's the most abundant, which is obviously what many other people have found as well. And just the color codes indicate here, this is for the cDNA data, the level that in terms of abundance, relative abundance of viral reads at different points in the time course, and also different cell lines. And so you can see the most abundant one is in the Vera cell line at 24 hours. And so, so there's the N off, the ORF7A and M, and there's the three most abundant transcript products. Then we saw something a little bit unexpected. And the fourth most abundant isn't what would be called a canonical subgenomic mRNA. It's a little bit different in that the 5' section doesn't actually end in a leader sequence. It actually trails on into the first non-structural protein, ORF1AB. And so therefore the annotation that gets is leader ORF1AB, comma, ORF10. So the disjunction after it finishes in the ORF1AB gene, there's a disjunction which goes almost right to the end of the molecule into the ORF10, which is not really thought to be a protein coding ORF. And I'll come back to that later, and then goes to the end of the molecule. And we see another one of these at the end here, this leader ORF1AB N. So it also jumps out of the ORF1AB and then has a disjunction to the just upstream of the N ORF. I should say that this annotation, the name of the ORF which appears after the comma, reflects the next ORF, which is downstream, 3' downstream of the junction point, because it would need to include the start codon in order to be able to produce that molecule in frame, obviously. And so, based on the fact that we clustered reads into read isoform into isoforms, we could then look at the depth of coverage for each of these classes. And so we could generate these plots, which show the depth across the RNA genome, so from 0 to 30 kb. We've just got the annotation of the genome up the top here. So for instance, the most abundant class was the N-ORF, as you recall, and so the depth profile of that is indicated with this kind of yellowish color here. And you can see that it has very high depth at which, as I say, just starts just prior to the dashed vertical lines indicate where the open reading frames begin. So the coverage starts just prior to that, and then runs to the end of the genome. And then you can't see it very well, but at the start, there's a bit of yellow at the same level of abundance, indicating that this includes the leader sequence at the start as well. And so those slightly unusual transcripts I was talking about, I should also emphasize, sorry, that the y-axis is on a log scale. So these unusual non-canonical transcripts includes this ORF1AB, ORF10 transcript is indicated here in pink. And so you can see that the depth of coverage starts to drop off a little bit into the ORF1AB. So it gets to, it includes the NS, almost always includes NSP1, so the first non-structural protein, before it drops off. And so here we are. And so it gets to NSP1, so we've got those NSPs indicated at the top before the depth profile starts to drop off. And then, so it's skipping this middle section, and then you can see it picks up again within the N-ORF, so not upstream of the N-ORF, but at some point within that, and then including basically all of ORF10. Yeah, so that's the non-canonical transcript which we identified there. And you see the other one here was this one which then joins up just before the purple, just joins up actually with quite a sharp junction here at the N-ORF. So we went back and looked at some previously published data published by Rigor et al. So this was a coronavirus 229E genome, which was also done using direct RNA nanopore sequencing in November 2019. November 2019 was published, so quite just before the onset of the pandemic. So we could also reanalyze that data using our approach. And so you see that actually the transcriptional profile looks pretty similar for 229E. And you also see these non-canonical RNA as well. In particular, this pink one also includes NSP1, and then also includes the 3' UTR. So this 229E doesn't have an annotated ORF10, so but instead it joins up just to include the 3' UTR of the molecule. So the next thing we looked at was the breakpoints. So just generating a heat map of breakpoints in order to see where the junctions occur. I say splicing here, I really shouldn't. It's this obviously negative strand discontinuous extension process. So this left-hand heat map here is showing where the breakpoints occur for the canonical transcripts that we were discussing before. So on the y-axis here, we've got the 3' end of the molecule, and you can see it's ranging from 1 to 100 base pairs. And most of the breakpoints are occurring around 66 or 66 base pairs approximately. And then they're joining up. So on the x-axis, you can see the 5' end of the molecule. And so you can see that, as we've seen before, the most abundant breakpoint position is between 64 base pairs here or 65, and just upstream of the N-orf. So that just, I guess, another way of looking at what we saw before. And then on the right-hand side, we've kind of zoomed out on the y-axis, if you like. So this is looking now at these non-canonical transcripts. And so you can see basically this left-hand heat map fits in right in this very top little sliver of this right-hand heat map. But we've expanded it out, and you can see there's a lot of disjunction occurring, say from 1 kilobase onwards down to 10 kilobases, but it's quite spread out, and that was reflected in the coverage plots having a gradual decline as well. And although it's spread out on the y-axis, it's less spread out on the x-axis where it joins again just upstream of so-called ORF10, which is not thought to be a functional ORF and really is really just the 3' UTR of the molecule. So if you remember, we were looking now at the subgenomic mRNA. We wanted to know what proportion of the subgenomic of the total RNA was subgenomic mRNA, and we were also interested in whether it was possible to use that as a measure to track the transcriptional activity of the virus in cells. So we looked at the proportion. So for each of these open reading frames, we looked at the proportion of reads covering that open reading frame, which also included the leader sequence, and that's what's plotted here. So we've got the different cells arranged from top to bottom. So Kako at the top, and then Kalu and Viro. I'll talk about the bottom right in a minute. And then we've got the different ORFs lined up at the bottom. And so you can see, for example, that, and then the time points as well. So if we just look at the Viro, you see in green the 2 hours, there's this ratio is 0.25, then it peaks at 24 hours and then gradually decreases again. And then you can see for the other samples as well, there's not much at 2 hours, but it does increase to its maximum of 24 hours in terms of the proportion of transcripts which are subgenomic mRNA, including the N. And then down the bottom here, we also sequenced RNA which was extracted from the virion prior to infection. So, and you can see the profile of that is markedly different, and that's reflecting the fact that there's not a lot of subgenomic mRNA in the virion molecule itself, and probably what we do find may be contaminant from cell anyway. So you can see here there's very little reads which include the leader covering these open reading frames. And so based on this, we sought to design primers which you could use to monitor the transcriptional states of cells without having to go through the expense of sequencing. So we designed primers which covered the N-orf, essentially, which would measure the total expression of the N-orf, and then primers which would capture the expression of the subgenomic mRNA, which had the left primer in the leader sequence and the right primer just downstream of the open reading of the start codon for the N-orf. And so based on that, we could then validate these observations using PCR. So this is showing the difference in the CT, the delta CT, between the subgenomic primers and the total primers over the time points. And we did this for the N as well as the E ORF. We included E because E is being used by other groups as a measure of transcriptional activity, but the N one we designed ourselves. And so you can see, well, firstly, basically as time increases from 0 to 48 hours, you can see that the delta CT decreases, reflecting the fact that there's an increase in the proportion of subgenomic mRNA. And we can see that the delta CT for the N-orf is smaller than the delta CT for the E-orf, reflecting, which reflects what we see in this as well. This is a high proportion of subgenomic mRNA with the N-orf and with the E. And that's consistent across the cell lines pretty well. So essentially, this is a pretty good way of tracking transcriptional activity of an infection within a cell. We've applied this since to looking at the new variants of concern. And so we see, so this is showing for alpha, beta, and delta. We're looking at the subgenomic N and the total N, subgenomic E and the total E. So we can see that the actual raw CT based on the total N is lowest in the beta variant. This is after 48 hours infection in vero SLAM cells. So that beta variant was grown to the highest titer. Then on the next slide, we can look at the delta CT. And so we can see that here, the delta variant actually has the lowest delta CT, both measured from the N-off as well as the E-off, indicating it at 48 hours, the transcription of this delta variant is higher than the other two variants of concern. I should emphasize this is just a single replicate and quite preliminary data, which we need to do some more work to validate. Yeah. So we also use the PCR primers that we designed to look in patient samples. So there's this kind of mystery, if you like, as to why some patients are long-term PCR positive for the virus. And we were interested to see whether those patients were also transcriptionally active, well, at least as we could measure by the subgenomic mRNA. And so we looked over a time course of various patients that we had access to at the delta CT and the total N. And so you can see here that that in fact it's very hard to see any correlations. I mean, so just look at one patient, for example. So maybe patient A, you can see that over the time course that's being measured here, the CT is gradually decreasing, so indicating the viral load in the patient's getting lower. And at the same time, the delta CT is pretty flat, or if anything, decreasing slightly. So it's a bit confusing in a sense that this would indicate that although the viral load's getting lower, they're still transcriptionally active in that patient. So others would follow more of a pattern that we would have expected, whereby the pattern we were looking for was that as the CT increased, and so the viral loads got lower, we thought the transcriptional activity would decrease as well. But and so in order to see if that was generally the case, we just looked over all the samples we had access to and the correlation between the CT and the delta CT, and you can see there's very little correlation. And so I guess we conclude from that total CT is not necessarily a measure of the transcriptional activity of the virus which is present at that time. So these were somewhat confusing results to us, not quite what we expected. But I think, so I think the delta CT is an interesting measure which may, and we obviously need to look in larger samples and look in more time courses as well. So as I mentioned, one of our goals, so now going back to the sequencing data, one of our goals was to do differential expression analyses of the different time courses. So this is looking at the vero cells. We were able to look at the differential expression between, on the left-hand side, the 2 hours and the 24 hours, and on the right-hand side, the 24 hours and the 48 hours. And so we see here what you might expect between 2 and 24. If you recall, vero had a quite dramatic increase in the proportion of reads mapping to the virus during that time, and that's reflective in a big switch on subgenomic mRNA, canonical subgenomic mRNA, as you might expect. So we see a lot of upregulation of this kind of standard canonical transcripts. We also see upregulation of these non-canonical transcripts, which include more of the ORF1AB, so particularly the first non-structural protein of ORF1AB, and then the 3' end of the molecule. So that's indicated in red on the left. So typically the canonical molecules are getting switched on in the first 24 hours. And then in the second 24 hours, we see something quite different. We see that the canonical mRNA are actually downregulated. So they're getting kind of switched off, if you like, to a certain extent. And then we see an increase in these RNA fragments of RNA, which doesn't include either the 5' or the 3' end. And we also see a downregulation of this non-canonical ORF1AB, ORF10 molecule. And then in the other samples, we couldn't do a comparison with 2 hours here because we didn't have enough data for 2 hours for these human cell lines. We could look at the changes between the 24 and the 48 hours. And again, we see between 24 and 48 that the canonical mRNA is downregulated, and some of these fragments are upregulated. But also here we see the upregulation of this non-canonical subgenomic mRNA, and we see, some similarities, at least in the other cell line as well. But then trying to summarize that and see what's going on in terms of expression of canonical versus non-canonical subgenomic mRNA, we summed up all of the canonical mRNA reads and the non-canonical subgenomic mRNA, and we could see that, this is summarized here, we could see that at 24 hours, the dashed line indicates the canonical. So another word for that would be TRS dependent. So it's basically formation at the expected TRS positions. So we can see that that is switched on a lot between 2 and 24 hours, as you might expect, because the virus needs to produce all of these proteins, and then that drops off at 48 hours. And then on the other hand, so the solid line down the bottom indicates the TRS independent. For Vero, that also gets switched on in the first 24 hours, but for the human cell lines, that seems to be delayed somewhat and continues to increase between 24 and 48 hours. And you can see, of course, that the overall abundance of this TRS-independent subgenomic mRNA is substantially lower than the TRS-dependent subgenomic mRNA. So just going back to some of these fragments, so in particular, we saw them in the Vero, we see these fragmented mRNA, which doesn't include the 3' end. And that was a bit of a mystery to us because in principle, we shouldn't have been sequencing them at all because they're not, in principle, they shouldn't have the poly A tail. So we looked at these different classes of subgenomic mRNA. So whether they had the 5' end and the 3' end, so that's down the bottom here, so that's like normal subgenomic mRNA, or whether they just didn't get through to the 5' end, and that's also consistent with just not sequencing the full molecules. But the ones we're interested in were really the top line, so the no 5, no 3, things which didn't get to the 3' end and also didn't get to the 5' end. And so we can see here that most of those molecules, at least in the majority of these samples here, don't have a poly A tail at all. So this is using nanopolish to see which molecules have a poly A tail. And so the blue color here indicates that they've got the poly A tail and the adapter. And then that's what we expect to be sequencing. But you can see that we're actually sequencing quite a lot of reads that don't have a poly A tail. So I guess the conclusion from this is that a lot of these, we're still sequencing these fragmented molecules, even though we're not really targeting them. They still somehow, many of them manage to go through the pore, despite not having much of a poly A tail. Except for the 48 hours, there seems to be some polyadenylation of these molecules, and that's something we're looking into now to try and figure out why they're polyadenylated and how long those tails are. The other curiosity we saw, so our clustering algorithm didn't restrict to subgenomic mRNA which had a single disjunction. So it was basically the way that the hashing worked, it was possible to pull out of their reads which had multiple disjunctions. And indeed, we saw some of those, but admittedly at quite low levels. And this is just an illustration of what these double junction subgenomic mRNA look like. If I could perhaps draw your attention to the yellow one here. So that's been annotated as leader and then a break to the M. So the first disjunction going from the 5' to the 3' end is between the leader, and that looks like a normal canonical junction, if you like. It's just a very sharp break just upstream of the M open reading frame. But then these molecules stop, well, have another second disjunction with downstream of the M ORF, and so it misses a bit of sequence here, and then it joins up again upstream of the ORF10. And so as we saw before, many of these non-canonical mRNAs had their junction just upstream of ORF10 and 3' UTR. So in this case, these are normal sgRNA, which have a second disjunction upstream of the 3' UTR. We can look at the abundance of these double disjunction molecules down the bottom here. So we see them both from the cDNA library prep as well as the RNA library prep, and we see that they have abundance profiles which are consistent as well. So, for example, they're highest in the CALU 48-hour time point. So they do seem to increase during the course of the infection. And it's not, of course, the process by which the subgenomic mRNA is formed during the replication, as you recall. So it's not impossible, of course, that it could have multiple events where it forms a disjunction as it's forming that negative strand. So as I mentioned before, we also sequenced RNA as well as from all the cellular infection models from the virion itself. And we looked at the error profile in terms of the base calling error relative to the reference genome. And we noticed that the virion molecule had a higher error rates and quite markedly different error profile. So this is just showing across all of the open reading frames, what's the error ratio or the distribution, so the distribution of the error ratio. And so you can see for the cellular derived SARS-CoV-2 mRNA that the error profile is quite tight, or relatively tight, around say 6%. But then for the virion molecule, it's consistently a few percentage points higher. So it's a very striking difference in the error profiles, base calling error rates. And of course, one explanation for this is that there's different RNA modifications on the RNA which we extract from the virion versus the RNA that we get from cellular infection. And as you recall before, I showed that there's very little subgenomic mRNA in the virion model. So essentially this is measuring the error ratio of the RNA genome compared to the subgenomic mRNA. So we then looked to see what the differences were in RNA modification between the genomic RNA, which we extracted from the virion, and the subgenomic mRNA as well. So just to explain this figure, basically these are lined up here. Sorry, just trying to get here we go. So these are lined up primarily according to the cell type. So at the top row, we've got the CACU, and then we've got the CALU, and that moving down to the viro 24 hours, and then the viro 48 hours. So these are the different cellular infection cells, and also 2 times points for viro. And then within that, we've got these different subgenomic mRNA. So using the read clustering approach, we could cluster reads into ones which had the NORF subgenomic mRNA, and go back and extract the actual raw data in order to look at these RNA modifications. And what's been plotted here is the difference, I should say, sorry, we use nanopolish to estimate the frequency of RNA modifications within each of these different classes of subgenomic mRNA. And then we also estimated the proportion of RNA modifications on the RNA genome. And what we've plotted is the squared difference in the fraction of methylation between the specific subgenomic mRNA and the RNA genome. So the RNA genome is just a single data set, that's constant. What's changing is the subgenomic mRNA that we're comparing it to. And so we see that we saw 3 areas with quite substantial difference here, and so they're indicated as C, D, and C, B, and D, and that has a negative. It's a squared difference, but we keep the sign. So negative indicates that the subgenomic mRNA is less methylated than the RNA genome, and so yeah. And we don't see really many examples where the RNA molecule subgenomic RNA has more modification than the RNA genome. And the other thing we noticed is that it's very consistent across the subgenomic mRNA. So yeah, that's indicated by the fact that the lines are solid and they don't really drop out as you move across the subgenomic mRNA. So that indicates to us that at least most of the methylation that we're seeing, or modification that we're seeing, is on the RNA genome itself. rather than the subgenomic mRNA. Although, of course, it doesn't rule out the possibility that there's subgenomic mRNA-specific modifications, just that we didn't see it using this approach. And then on the right-hand side, you can see what the raw nanopore signal looks like. So it's this squiggle pattern. And so you can look at the difference between the top and the bottom to see that there's very subtle changes which can be picked out by nanopolish. But I guess it's most evident perhaps in G here. You can see that there's a shift around this position here, shift upwards in the signal, which we say is likely due to the presence of a modification. Okay, so I will finish there. Just to conclude, so direct RNA sequencing, I think, provides lots of insights into transcriptional activity in SARS-CoV-2. In particular, I guess, uniquely, it can look for modifications in RNA. We can look at the different categories of subgenomic mRNA. We can also look at polyadenylation. So in our analysis, the epitranscriptome seemed pretty stable across the time points and across the different subgenomic mRNA, and also the different cells as well. But it did seem to differ from the RNA genome itself. So we infer that the RNA genome has stronger RNA modification, but the transcriptome obviously is much more dynamic. So we've developed a new software package, which you can use if you like. And also we've made a shiny web server for people to look at the transcriptional profiles in our data as well, which is indicated there. So this is a big team effort from many people. So I should say Jesse Chang led a lot of this project from the lab perspective. Daniel Rawlinson developed together with me a lot of the bioinformatics. Miranda was involved in, did a lot of the sequencing as well. And we also relied hugely on support from the Superbug group in terms of infecting the cells with SARS-CoV-2, and also colleagues from the Victorian Infectious Disease Reference Laboratory for providing the infectious sample, and also help from the Clark lab particularly in the sequence and the PCR cDNA. So I shall finish there and happy to take questions. Thank you.
Yeah, Lachlan, thank you so much. Very cool stuff, and that was a ton of work. Audience, please either type your question into the Q&A box and or raise your hand and we'll call on you. I'll get things going. Lachlan, I just had a really basic question. I missed how are you enriching for the things with poly A tails? You're just running on an oligo DT column, or what? Why are you so surprised?
Oh, I see. So direct RNA sequencing, the adapter basically is designed to attach to the motor protein. Everything's designed to attach to the poly A end. And then, so for direct RNA, the molecules without a poly A tail shouldn't, in general, have that adapter. So therefore, it shouldn't pass through the pore. But I guess, stochastically, it could probably attach. And we're not really sure what the length of poly A that's required. So maybe there's just within the sequence a small, like a couple of A's, and it can join there. And for the cDNA, it's basically the first step, the primer, which is used to amplify, has poly T. So you do the first transsynthesis, and then there's a poly T primer to amplify from that end. So as well, it should have the poly A tail. Okay, excellent. I have no direct experience with nanopore sequencing, so I found the RNA base modification really intriguing. So I Googled and saw some papers that this other group is certain that there's 2' O methylation at some bases, particularly at the genome cap. What other RNA modifications do you guys have, current profiles for? Right. Yeah. So we're not necessarily able to find all of the RNA modifications, just the ones which result in a signal difference in the squiggle plot, if you like, as the molecule passes through the nanopore, and if it changes the electric current, and then they don't all do that, and they do that in different ways. So we've just been using nanopolish as like a catch-all way of looking for differences in that signal. We haven't really paid a lot of attention. We probably should to different classes of RNA modification. We think that M6A is particularly well captured here, and 7mg, but we're not sure that's so important. Yeah, at one point we were digging into the different types of molecules, but it just does get quite messy, and so we just decided to do a kind of catch-all approach for now. But there are a lot of tools out there designed for looking for specific changes, particularly M6A.
Yeah, is it thought? Is there a theory why the virion RNA would be so heavily methylated? That, I mean, the idea that popped into my head was, something like, how heterochromatin gets heavily methylated and it helps the chromosome collapse. So I didn't know if it would pack better inside the virion if it was methylated and maybe more collapsed.
Yeah, and like you, I had that thought as well, that it might be to do with the packing of the molecule. But other than that, I haven't really thought about it that much. But yeah, so there's something going on there. There's definitely a big difference, possibly it's to do with packing. It's a really good question.
Another basic question that reveals my ignorance about nanopore sequencing. The 7 to 9% error rate, are those errors generally stochastic, or are there patterns that are more likely to produce errors?
Well, certainly in the presence of modifications, there are errors which are more likely to be produced. And also one of the main error modes for nanopore sequences, homopolymers. It's hard to get those accurately, and they can create insertions and deletions. One of the blessings and curses of nanopore at the same time is that the software for base calling is constantly improving. So probably if we were to redo this with the latest version of Guppy, we would push that error rate down a little bit. I mean, the process of base calling uses neural networks and continually is kind of updated from more data. So yeah, but the main error modes are insertion and deletions.
Okay, and that's the when it gets hung up in the pore and it gets called.
Right, might have slightly longer dwell time in the pore, and that leads it to insert an extra base or something like that.
Yeah. Oh, such a cool technique. Hey, I had a question for this may be outside your purview, but what do you think it is about the Vero 6 cells? Why are they so good at being infected with this virus?
Right. Yeah, we've looked. I didn't present it here. We've looked a bit at the host response changes as well. My understanding is they're defective in the interferon response. Right. So they're missing a key part of the immune response there, and that allows entry. I think that the Vero H SLAM as well, as you say, it's not really my area of expertise, but I think that has a extra genetic modification which may enable the virus to enter the cell more effectively too. I'm not entirely sure.
Do you guys see changes in the viral genome after passage through these cell lines?
Yes, we did see that. We saw, to really describe it here, but we saw some deletions in the 3' UTR after passage through the cell line. Yeah. We should have a look. So we've only looked at that in the original Wuhan strain. We should have a look in some of these newer variants to see if we see similar deletions in the 3' UTR. Yeah. There's a suggestion that might be adaptive for the cell culture.
Yeah, I mean, I've read that it loses the polybasic cleavage site in passage in Vero 6. Do you guys see that, or?
We'd have to have a closer look at that.
Yeah, I mean, maybe that's longer serial passaging before it loses that. I don't know. This is just something I saw online.
We didn't see that in the Wuhan strain, but we should look again at some of these new variants that we've used. It's a really good point.
Yeah, that's really interesting. The shift in the subgenomic RNA distribution over time. And then I thought it was really interesting that, I just had this naive picture that, of course, virion RNA is just going to be a single copy that just starts at base 1 and goes to base 30,000. But you're right, there's no reason to believe that would necessarily be what's packaged.
Well, I'm not sure that necessarily shown that's not the case.
No, just to even look, right? Just to even look that could not be the case. That there could be subgenomic RNAs.
It should just get packaged up, perhaps. But yeah, we don't really see much, and probably we do see some subgenomic mRNA in the virion, but at very low levels, and it's probably contamination from RNA. It's hard to completely isolate the virion.
Yeah.
Without any RNA.
Yeah, it was just a cool thing to look for.
Probably more naive than anything else. We're just sequencing everything we could get our hands on.
Yeah, sure. What are you guys going to work on next? What are the unanswered questions in this area, do you think?
We're trying to look at this from a single cell perspective at the moment. Mm-hmm. So, we've got a project where we're looking at infecting an organoid model and looking at it from a single cell perspective. I mean, in terms of unanswered questions, I think the RNA modifications is something that's in terms of exactly what they are and how there perhaps is subtle changes that we haven't really picked up so far. Some of the points you raise about perhaps changes that might occur during cellular passage as well. Mm. They're the things that spring to mind.
Yeah.
At the moment,
Yeah, I mean, the technologies that this nanopore sequencing is just getting better, so much better, so quickly.
Yeah, definitely is improving for sure. Yeah.
Okay, well, Lachlan, thanks for your time. That was really amazing.
Thanks for the invitation. Thanks everyone who stayed through the end of the talk. I appreciate it.
Yeah, and sorry everyone, we don't have a slide, but join us next week. I believe it's also at the later time because we have another Australian. Okay, stay safe. Bye-bye.